Can you hear me if I just talk loudly? Mm-hmm. Yeah, at the back. Okay. So my name is Simon Gane, and when Emma and I were talking about what we were going to do today, we divided up in a way that doesn't really happen in real life, the difference between in the perception of coffee, which Emma's been talking about, between gustation, the perception of what happens in your mouth, chemoreception and taste, and olfaction. And so I am Simon Gane, as I said. I'm an ear, nose and throat surgeon. I'm present at the Royal London Hospital. I will be moving to the Royal National Throat, Nose and Ear Hospital, which none of you will ever have heard of because it is the world's, I think, only standalone ENT hospital. It's very small. And I was starting there in July as a consultant. And I'm also a PhD student. So my PhD is in a very weird theory of olfaction, which talks about sort of a quantum biological approach to how those receptors that we were talking about recognize olfactory molecules. I'm not going to bore you with that, don't worry about it. What I want to do is give you an overview of something that might be a little bit more interesting to you about what actually is going on when we're sipping coffee and appreciating it in the ways that Emma was talking about. So just full disclosure, my favorite coffee brew is AeroPress. I'm particularly fond of Ethiopian beans, so I thought I'd just get that out there. And I think to start, hopefully that chimney is worn off a bit. So to start, what I'd like you to do is you get the tubs. There are two sweets in there. You get the jelly bean out. Hold your nose with your other hand. Everyone's got two hands. Pop the jelly bean into your mouth. Holding your nose all the time. What does it taste like? Not like much. Have a chew. Now, let go of your nose. And suddenly, well, for me anyway, suddenly the taste of jelly bean comes in. You know what flavour it is now. Yeah. I'm sorry. Strike one for the jelly bean. So that is meant to show you what the power of smell is in our perception of flavour. We talked about these six putative flavour detectors on the tongue and in the mouth, the basic tastes, plus the other things that go together with your perception of flavour. But the power of smell is probably the most powerful and most detailed part of our perception of flavour as a whole thing. So what are we looking at when we look at the sense of smell? Well, this is hopefully some of you have seen it. It's a person called Compound Interest, follow on Twitter. They made a poster about the chemicals that come off coffee when you come. You can download this for free. It's a Creative Commons license poster. It's very interesting. I want to draw your particular attention to a couple of molecules. 2-furfuryl thiol, the first one up on the top there, to the left of the screen. That's the classical, canonical smell of coffee. I think it's a tragedy to call that the smell of coffee. The nice thing about coffee is its complexity and the depth of aroma that it comes from. But that is usually identified as the thing that smells like coffee. But you can see there are a whole load of other things coming off there as well. The other thing I'd like to point out is guaiacol. I'm not going to tell you why, but just if you can remember that molecule for later, it has quite an important role to play. This is sort of smoky, spicy scent. I actually can't perceive guaiacol too well, and I couldn't perceive the PTC either. So what's happening when you take that first sip of your first brew of coffee in the morning? I want to take you on a little molecules view of the nose via the power of nasendoscopy. So this is something that I do every day. Aren't you glad you don't have to do it? This is putting a rigid nasendoscope, actually a flexible nasendoscope, which is a flexible fiber optic tube in through the nose and down through the nasal cavity, which goes all the way down to the level of your ear, into the postnasal space, which is the top of the gullet, swallowing tube behind the nose, and that's looking down onto the larynx, the voice box. You can see we're going to do it again. So first of all, septum down this side, the cartilaginous division between the nose, the little shelves of bone that line the side of the nose called the turbinates, that's the inferior turbinate on the left of the screen there. Through the posterior carina, that's the postnasal space where the eustachian tubes drain from your ears, and then you're going over the back You're looking at the tongue base, the little funny little thing there is the epiglottis, and then those two little things that are closing together, those are your vocal cords. And that person was actually phonating. They were going ah. They couldn't articulate. They weren't making a voice, but they were going ah. And you can see just at the end there those vocal cords moving. What this doesn't show is what the molecules are doing when you sniff, which is slightly different. Normally, when you're breathing, this is the way the molecules are going. You're going over the back, down the throat, through the voice box, into the air, into the lungs. But when you sniff, something different happens, and a larger proportion of the air is diverted upwards into the top half of the nose, and that's important because that's where the sense of smell lives. So this is called a coronal CT of the head and sinuses. So you can imagine it like a loaf of bread, slices going all the way back through your head. You can see the eyeballs there. These are your sinuses. There's a nice, well-aerated face. You can see that. Sorry for those of you who are not used to looking at CT scans. White is bone, grey is soft tissue, and black is air. So you can see that in the middle of your face, you have a whole big airway going all the way back through the nose. Sinuses on either side, sinuses at the top, sinuses behind. But the important thing is here. At the top of the nose, outlined in red there, the lining of the nose changes from what we call respiratory mucosa, or epithelium, you can think of it as skin, to a specialized form of epithelium called olfactory epithelium. And it's specialized because of a number of different things. It's covered by a different type of mucus, which has compounds in it which process the odorants that might be coming in, compounds that bind them and move them to the receptor, and loads of other things, probably protective as well. And then just within that mucus, there's the olfactory sensory neurons, or the nerve cells that are direct extensions of your brain. Unlike almost every other nerve in your body, they are directly coming from the brain into the nose, which makes it fascinating for a number of research things. But more importantly, this is where you're going to do the detecting, right up at the top of the nose in the olfactory mucosa. So it's easier probably to look at it from the side, if you will. So instead of looking at it from front on, we've now done a slice that way. It's called the sagittal view. And this is so you can see the nose pointing off to the right. Those little crenellations, the black bits, those are your sinuses, some sinuses. See some grey blobs in the middle of the nose. Those are the inferior turbinates coming through. But what you're doing there is looking onto the lateral wall of the nose. It might be easier to see it in a sort of diagrammatic format. So we're looking onto the side wall of the nose. So you're sitting in the middle of the head and you're looking onto the side, out towards the eye or the ear. And here you might be able to understand a little bit of what's going on. So say you get your cup of coffee, this abomination that is the lid, and you're sniffing it up, and it's going up into your nose from the front. This is called orthonasal, straight nasal olfaction, coming from the nostrils, what everybody thinks of as sniffing or smelling, into the nose. But the weird thing is that I got you to hold your nose at the front, but you put the jelly bean in your mouth and smelt it some other way. You're holding your nose in the front. That stopped all airflow through the nose. But as soon as you opened your nostrils, you weren't smelling it out your mouth was closed. Not from you. The other ones, what you were doing is called retronasal olfaction. So the coffee goes into the nose and it comes up the back through that nasopharynx that I was showing you earlier, the top bit of the gullet, up the back into retronasal olfaction. And that's where the whole percept of flavour comes from. You're actually smelling what is in your mouth, bizarrely. Some people find it disgusting, but that's just what's happening. Those molecules are finding their way, you're mashing them up. There are enzymes in your mouth that dissolve molecules and turn them into volatiles, which can come up into this postnasal space and retronasal olfaction. And this might have something to do with something that's bothered me for a long time, which is the number of people who tell me how much they love the smell of coffee, but they hate the taste. Why is the taste of coffee so different to its smell? I still don't understand. Emma has a good theory about this. But yeah, what they're still doing is smelling. It's just through the back of the nose rather than the front. Good. So how are they doing the smelling? What's going on? So as I said, there's a specialized mucosa lining skin at the top of the nose with nerves projecting into it, direct extensions of the brain. So you can see these little greyish things are our nerves, and they're bundles of olfactory sensory neurons, which are specialised nerve cells that look a bit like Medusa. They've got this bulb at the end with these cilia projecting out, these hair-like projections of the cells, and they stick out into the olfactory mucus. And their whole job is to, like an anemone, grab olfactory molecules and recognise them. So they're sticking out into the mucus, and on their surface is a specialised protein called the olfactory receptor. Now, olfactory receptors are really clever. We don't actually know that much about them. It's what my research is largely in. And I'm not going to bore you too much with the physiology. I don't know how much you know about neuroscience, but basically they're activated by some way. They cause a cascade of, every time a receptor is activated, there's a cascade of proteins changing within the cell that causes charged ions to cross the cell membrane, that depolarizes it, which in other words you can change of it as suddenly, you can think of it as suddenly switching on the electrical activity. That charge rushes up the nerve all the way up to a specialized structure at the base of the brain. Inside the skull, they run through this tiny little sort of sieve-like bone called the cribriform plate, up into a specialized structure called the olfactory bulb. And the olfactory bulb is really where the clever bit starts. So the problem with these olfactory sensory neurons, they're direct extensions of the brain going right into your nose, and they're constantly, unlike any other part of the brain, exposed to viruses, snot, dirt, asbestos, whatever, coffee, whatever you're putting into your nose or breathing up your nose. They're constantly under assault, and they're constantly dying off. So it's one of the only two places in the whole of the central nervous system where the cells regenerate. Imagine if you could regenerate any other nerve. You regenerate after a spinal injury. You break your back, you paralyze for a bit, and then you start to walk again. It just doesn't happen. Although there's a lot of research going on looking at the properties of these nerves to regenerate, and at least one person is able to partially move his legs because of the research going on about olfactory sensory neurons. So the problem is that they're constantly regenerating, they're constantly growing up to the brain, but the brain has to stay still. It can't afford to change shape, because that shape is how we encode experience, how we encode memory, how we encode who we are, the relationship between those neurons. So it doesn't change shape. And the olfactory bulb is the way that the brain solves this problem. So your nerve is going up into the olfactory bulb, and here's a sort of a simplified diagram of this. And this is what happens. If you have an M molecule, so each of these olfactory sensory neurons expresses a particular receptor, and about 400 different receptors that it could choose from. In your DNA, you've got the recipe for about 400. It picks one and expresses multiple copies of only that one receptor. It knows what it's going to do, and say let's, for example, the name is not important, but let's say it's the M receptor. So on its cilia are all these M receptors, and the M receptors only recognize a certain molecule or a certain number of molecules. So when an M comes floating in, it lights up those receptors, lights up that nerve. Another one comes floating in, that lights up that nerve. And all of those nerves send their signal up to the bulb, to the same place within the bulb, a structure called the glomerulus, an olfactory glomerulus. You can think of it as just a ball of cells, really. All of these cells are synapsing within this glomerulus, they're joining, they're sending their signals up, and then one single strand or nerve, well, multiple nerves, but you can think of it as one line goes from that up into the brain. And it's quite clever, actually, because say you've got an E receptor coming in as well, so that does that, an MRLOT, that's that activation pattern that gives you the sense of a Merlot, the spatial activation of those different glomeruli, and therefore the different receptors, because remember, each receptor goes to its own glomerulus. Tells you this, that's what those molecules are going, that's what Merlot is. But it may be that you're not sniffing Merlot. You're sniffing something else. There's definitely an O, there's definitely a T, there might be an M, a P, an I, and an O in there. Well, it's not Merlot, it's Pinot. And that different activation pattern is what your brain recognizes as the smell of Merlot, Pinot, Yirgacheffe, whatever. So that, because those glomeruli are fixed in space, they're constant, the brain can then wire up to them to know that activation pattern means this thing, means Emma was talking about a perception, means the sense of this wine or that coffee or that food. They're all then wired up to further bits of the brain, which go into, so the olfactory bulb goes to something called the piriform cortex. That connects to other bits of the brain, like the thalamus, which is very closely associated with memory and with emotion. So famously, Proust's Madeleine evoked this entire, 6 books of turgid prose. But that's where that's happening. That feedback from your memory and from your emotion is being evoked. But not only is it being evoked by the smell, it's also via that and something called the orbitofrontal cortex feeding back to the piriform cortex. So Emma was saying about how your expectations, your memories, your socialization, your culture all feed back on your perception. They don't just make you think of things in a certain way. They actively change the way you perceive certain smells. And there are MRI experiments looking at this, and they've shown that the spatial representation of activation within the piriform cortex is what we perceive as a smell. So you can actually tell for only one person at a time. So your activation is not the same as mine, but if you can tell what somebody is smelling because the same neurons light up every single time. So that's how your brain is doing the smelling that it's doing, and that's the neuroscience behind what's going on. But my interest is really in the receptor, because all of this wiring is based on input from the receptor, about the information that you're getting at a molecular level. This receptor can recognize certain things and doesn't recognize other things, and it's quite difficult at the moment to tease out what that is. And I want to talk a little bit about the receptor. So the next experiment, you should have one sweet left. This one you just have to pop into your mouth. This is a Parma violet. So how many of you don't think this tastes so much? It just tastes as sweet. No? Yeah? Just sugar. Yeah? How many of you get a really strong floral violet flavor? So this is genetic, as with the bitter tasting on the PTC, but it's much simpler than the bitter receptors. So what you're smelling or tasting or not tasting is this compound called beta-ianone, which is the smell of Parma violets, classically the smell of Parma violets. And it turns out that what predicts whether you're able to smell this or not is a receptor called OR5A1. Again, the main picture doesn't matter. But what's so interesting about this, so this is a description of the receptor known as a snake plot. So you can imagine that below the grey line is within the cell, above the grey line is outside of the cell. Just one little substitution in the genetic code, one base that goes from an A to a T, changes your entire perception of Parma violets. So just one mistake, this is called a single nucleotide polymorphism, just one little mistake in the genetic code completely changes your perception in terms of the threshold, in other words, how strong the concentration needs to be for you to perceive it, changes the valence, how pleasant you find it, just in one receptor. And we've looked at a lot of receptors in mice, showing that receptors make a difference in human perception of taste and smell is more difficult. We've definitely confirmed 5 of those over the last 9 years or so. Remember I said...
only 5.
Well, there are 2 more. I think we've shown 2 more. But yes, out of 400. Yeah. So this it's fairly tricky to do. You need to basically find what's called a specific anosmia and show that that's related to the genes. We can talk a bit about that if you want. That's my thing. But what I wanted to point out to you again, remember I asked you to look for guaiacol, to remember guaiacol? There are 2 things that we do know definitely affect detection and valence. and that's isovaleric acid and guaiacol. So of the 5 or 400 that we know, isovaleric acid and guaiacol really do affect the human perception of these olfactants. So that's going to be important later on. And it comes to a bit of a sort of philosophical thing really here. So about 40 years ago, even longer now, a philosopher called Thomas Nagel published a paper called "What Is It Like to Be a Bat?" And his argument was that although we have sonar, you all know what sonar looks like. It looks like the little the submarines use. We can use it. We use ultrasound every day for looking at neck lumps, at new babies, all those kind of things. But although we can appreciate what it might be like we can see sonar by these things, we can't really ever truly know what it is like to be a bat, to not have that good sight, to not have colour vision, instead to see the world in reflections of sound. And he insisted that there was an unknowable difference in the qualia, in what it was like to appreciate these things, that meant that we couldn't ever truly know what it was like to be a bat. And so this brings me on to Emma's flavour wheel. So I love this. I think the design is amazing. The colours are beautiful, and I'm sure will be very useful. But there are some caveats that I have about it, not to be too contentious. So what we know, let's talk about a simpler system, right? So I said there are 400 olfactory receptors. Well, what do we know about vision, for instance? Well, there are only 4, really, maybe 5 vision receptors. You've got the dark and light receptors, the rods, and then the cones in the 3 colours that you know about. 3 colours there. Actually, there are probably 2 different types of blue detectors. And since they're carried on the X chromosome, a small number of women actually have 4-colour vision because the 2 types of receptors have slightly different signal intensities that they can respond to. But because you need 2 X chromosomes, only women can do this. Men, you're out of luck. So we know that just the mixing of 3 different receptors gives us this amazing gamut of colours that we can perceive, right? It's amazing. I mean, the detail that we get just from the colour variation, and these are just little different photons of different wavelengths and different intensities and different numbers that give us this variation. Over 4 receptors give us that, and the richness that we have in our visual world. Well, what do we know if we lose one of those? So there's highly likely that at least one man in this audience might not be able to see the colour. Can anybody not see a number there? Slightly uncontrolled because of the panel. Everyone see 27? So we know that it's actually quite common that men especially have the red-green colour blindness. It's called deuteranopia. And you can do experiments like this to try and make it show people what it must be like to live without the perception of the difference between red and green, this kind of slightly more yellowish, greenish tinge. Of course, that's not really what it's like, because the people who have colour blindness don't really know. We can't know what it's like to be colourblind, we can guess, and they really can't know what it's like to be fully trichromatic. So what does this mean for olfactory receptors? Well, as I said, there are 400 of them. And if you try and do a sort of bar graph, so this is based on some work from late last decade, and it was very limited. It was a lot of hard work back then when the sequencing technology wasn't as good as it is today. But you can imagine doing a bar graph. You take the receptors that you know, and you can see, if you look at their sequence, which ones will code for genes that work and which ones will code for receptors that work and which genes will code for receptors that don't. So you take, I think that's 20 there, and you can read them out. So the red ones, they don't work. They've got a mistake in the code that means that whatever they produce, it will not be a functional receptor. Either that it's too short or it gets malfolded in some way, but you know that they won't work. The green ones are different to the ones that you know are functional but may be functional, and the dark green ones, they're definitely good for go. You know they're going to make loads of those. So you can imagine that for every individual you could produce a bar graph like that. You can produce a barcode of their nose saying these are receptors that work, these are receptors that don't. You can do that for 1, you can do that for 2, you can do it for 3, you can do it for hundreds. And what Menashe and his co-workers did is they looked at 187 people, they bar-coded all of their noses, and don't forget, this is not 400 receptors, these are just 20 that they happened to look at. What they found is that in 183 people, there were 157 different unique noses. 157 different patterns of genetic inactivation just in the genes. We're not talking about whether they're expressed in the nose, whether there are other problems with trafficking, nothing like that. 157 different genetic patterns of olfaction. And this is American. They tested on African American and people of Caucasian descent, and there's quite a difference between the two ethnic populations. And further work has shown that this actually is probably very variable throughout the world. And so this brings me to in Indonesia there's a tribe called the Juhi, and they have probably the most advanced description of olfactory language that we know. They have direct, in the way that we can say red, crimson, carmine, and green, but apple green, all those things. They have essential descriptors for smells. But no one's done this, but my bet is that the only reason that they have such a good and descriptive language for smell, and they don't run into the problems that Emma was talking about, where you have these panels, they need to be trained, and blah, 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 is because they have essentially the same genetic nose. Their receptors are all pretty much the same. We've been accumulating mistakes in our genetic code probably since the advent of trichromatic vision. Some people disagree with that, but because we don't rely on our sense of smell, they're not under evolutionary pressure to keep going. We don't. If one stops working, it doesn't mean you fall over and die. It just means your percepts are slightly different. So we're constantly accumulating errors in the code for them, and that's why everybody that you meet probably has a unique nose. So what does that mean? Well, if you take that color wheel just for coffee, remember I said to remember guaiacol? Guaiacol, there's a burnt, smoky thing down at the bottom. Isovaleric acid is named by name over the time. Probably what's happening is that every single customer who walks into your coffee shop and drinks your coffee is coming there with something missing from that wheel. Some little block that they're not perceiving, or that they're hyper-perceiving. This is the other thing. So those receptors, we think, some of them may not work very well, but some of them work better than the average, the so-called super tasters. So you might be getting people with a huge block. And although the wheel is very useful for talking amongst ourselves and being able to reference things in general, I'm not sure how applicable it is across the wider audience. And there is one final thing that I wanted to talk about, is that some people don't have a wheel at all. So about 1% of the population can't smell. They're anosmic. And this is usually the two main types: the congenitals, the ones who never have been able to smell, and they actually do quite well. They in the same way as nobody knows what it's like to be a bat, and deuteranopic people don't know what it's like to see red and green as different colors. They don't know what it's like to have a sense of smell, and they do just fine. They perceive the things in coffee or food or everything else that Emma was talking about. The warmth of the steam on their face, the slight bite of the heat in the mouth, the trigeminal reception, maybe a little bit of chili, the sweet, the sour, and the bitter that we tried earlier. All of those things are actually quite powerful in their perception of flavour. But it's the people who lose their sense of smell as an adult who really do struggle. And I work with a charity called Fifth Sense. They're quite new. They've only been going for about 3 or 4 years. And they're the only charity for people who suffer from anosmia. And I've been to a couple of their conferences, and obviously I see patients who have smell loss, and I've seen that it's actually devastating, even for people. I mean, imagine what it would be like in your line of work. I've seen chefs, I've seen perfumers, whose ability to perceive what they're serving has just gone out the window. And so some of them, it's absolutely devastating. But even people who are not working in a food or food service job find the loss of smell to be emotionally devastating. Now, there are a couple of reasons for this. I mean, they certainly all lose their perception of food and their enjoyment of food. But there are some indications that they also become less attached to their partners. The role of smell in sexual selection and enjoyment of intimacy, there's quite a lot of work going on about that. Interestingly, my favorite experiment is the T-shirt experiment. Have you heard about this? Mm. So the experimenters pay people to wear T-shirts, and then they bag them up and they give them to other people to sniff, and then they rate them on how attractive the sniffing is. And it turns out that women tend to prefer the smell of men who have a slightly different immune system, so a marker of a different immune system. They prefer their T-shirts than those who have a similar MHC. And there's something to do with sexual preference as well. Gay men prefer the smell of other gay men. Lesbians apparently don't have much of a taste one way or the other. It's just the way the science works. I thought that was quite interesting. And women tend to prefer the smell of other women, which I can understand. But these people are devastated by their loss of sense of smell because they know what they're listening.
What's the cause? A virus?
How long do you have? So yes, there are two main causes. Viral. So some viruses actually preferentially attack that olfactory mucosa, cause direct inflammation of the nerves, and if they wipe out the whole thing, then probably those regenerating nerves don't manage to regenerate back up. In the worst case, what happens is the nerves mess up their attachment to the glomeruli, and there's a global activation of the olfactory bulb, which is just a horrible smell. It's a burnt kind of, it's very difficult to describe, a burnt and rotten smell, probably because burnt and rotten things have a lot of different molecules coming off them. So our brain interprets a wide activation of the bulb, this is my guess anyway, as an unpleasant thing, because unpleasant things give you lots of different aromatic chemicals. So viral anosmia, post-viral anosmia, and trauma, head injuries, hitting on the back of the front of the head. Those little fibers that I was showing you, they're very, 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 very delicate, and the brain is essentially suspended in a bottle of fluid in your brain, in the skull, and it's surrounded by a shock absorbing fluid called cerebrospinal fluid. But if you have a rapid shake, the brain moves within the skull and tears off those fine fibers. And again, if they don't regenerate, it's tickets for the sense of smell. There are lots of other reasons, but those are the commonest. Unfortunately, some people, if you're lucky, you lose your sense of smell because your nose gets blocked for various reasons, because those we can usually fix. But the difficult ones are the other way. So the thing that I want to leave you with, and the thing I like most about Fifth Sense, the anosmia charity, is that they are really not about moaning about losing their sense of smell. Yes, they want to get it out there. Yes, they want to be there for people who are devastated by losing their sense of smell. But what their main goal is to make people enjoy, as Emma said, the gift that they have already, this ability that we have to appreciate this complex aroma of multiple molecules coming off, the interactions between the different things, to enjoy it while you have it, to not throw it away and just ignore it as most people do. I mean, I'm preaching to the choir here for most of you, but I think most of you will agree with me that in the general public, the awareness of the sense of smell and the pleasure that we can derive from that is quite poor, and their whole thing is to just the same as Emma said, to really enjoy the gift that we've been given of this sense of smell and the amazing properties that we have. So I just want to re-emphasize that. Whatever your unique nose, just remember that, and remember that we to enjoy your next cup of coffee whenever you have it. Thank you very much.